Thank you. Thank you for the nice introduction. I'll go ahead and share slides and let me know. Is that correct?
Yes, we can see it. Thanks.
Okay. So very quickly, I'm going to um, just copy in a link to a Google Docs. So this is it's a random idea I had. I have no idea if it will work, but if you have ideas during this talk, you can join a collaborative and post your ideas to a whiteboard. I'll keep this link active. You can add ideas. You could put your email. If you like the idea, you could join as a collaborator. Um, the success of this will determine if I do this in the future. And I won't really review them. It will just be there in existence, and you can just have some fun with it, see what's on people's minds. So this is the slide. So if you click on that link, you will see this, and you could just add your ideas there in each row. Okay, so thank you for joining this lecture. I'm going to talk to you about a topic that's near and dear to my heart, and that is tropical rainforests. I'll mainly focus on. I'll dabble a little bit in temperate rainforests, and this is a topic that I really invested a lot of my time in during my PhD, and it comprises almost half of the research in my current lab and over the years. And the idea here is just thinking about these complex forest ecosystems and how ecological communities use various strata of forests. And so this is really this idea of vertical stratification of animal communities, plant communities. And just to kind of give some context, we work both in the field, but we also have tried to scale these to a macro ecological lens. So you can kind of see the macro scale approach on the left that's very categorical and generic, and a much finer resolution of information on the right there in terms of terminology going from ground up to the emergent layer of the canopy. So during our field work, how do we actually do canopy science? We choose a tree that's safe to climb and a site that we think that a site that we want to sample in, whether it be frogs, reptiles, butterflies, ants, etc. We worked on a variety of taxa. We use these large slingshots. So I'm pulling down a slingshot there with a ball it's like a lead bag on a rope. We shoot over a branch up in the canopy, we rig our gear, and then we start the ascent up into the canopy. So this is just some video from some work in Madagascar. We were studying bird's nest ferns, this arboreal plant and epiphyte that's very common throughout the Asian Afrotropics. And so here I'm trying to get to that fern right there in front of me. I'll have some footage here in a second of a field assistant who is actually doing some of the surveys. So let me see here. So this is now footage from Krista Seidel, who is a field assistant.
Bird's nest fern up in the canopy.
So you can see she's hanging from a branch there. I don't know if the sound was activated when I share screen, but she's looking for frogs in this Asplenium fern. They're called trash basket ferns because they collect a lot of detritus from above and they just accumulate a soil layer. It's an extension of the ground, but floating in space. And now,
So
um,
here is an asplenium.
next one. Now Krista rigged too low. She tried to climb over that branch, failed, and so she had to restart her video. So now she's above the branch. She can actually get to the fern.
Yeah, at the bottom. Oh,
So you can
Hank.
see that leaf litter layer there. They're essentially sponges suspended above ground. And so they're little refrigerator units up there. They evaporate off water, cool the air, and serve it's an amazing little hotspot of biodiversity and in really a canopy environment that is akin to a desert scape, really, in terms of just availability of structure, depending on what system you're in. But you can see she's looking for frogs. They usually hide under the leaf, the termination of the leaf at the root ball. So she's and I also have some references there. I won't spend a lot of time talking about Asplenium ferns. It is a study system that is also near and dear to my heart. And just, there are some papers there at the bottom of the screen if you are interested in what we found. But, we are successful. Krista didn't find uh, a frog in that fern. But here's another field assistant. Nat Geo joined an expedition. Qian Li did some really beautiful photographs. And here's a frog that we found in that phytotomata, that tree hole there. And so it takes about an hour to do a standardized survey. We catch about one animal if we're lucky, especially at night. We repeat all of our surveys at night. So it's really bizarre feeling climbing these trees at nighttime. Daytime, at least you have some sense of where you are, and so it's a lot of effort for just a few data points. So it's maybe a frog per survey, frog per 2 to 3 surveys if you're lucky. Um, and so he caught that frog, put it in a bag, and tied it off to his harness, and then continued on his survey, looking in different crevices, microhabitats, et cetera, for whatever animal, ants, frogs, chameleons, et cetera. So today, what I'm going to do is in this kind of scope of thinking about verticality, ground, canopy, how animals are vertically stratified, the kind of the framework for my lecture today is thinking globally, regionally, locally, asking very similar questions and then discovering whether or not they apply. And so this is really a test of generality. And I encourage everyone to take this approach if you haven't already, especially PhD students as you're designing your research. If you have a question, push yourself to apply that question at different scales and it really, in not just space, but also in time. And we'll talk about temporal scales as well and how they interact with these different spatial scales here. And the reason I like this approach is that you often have these gradients at different spatial resolutions that are nested, and you can interact the different scales to test questions. And I'll get more to that in a minute, but this is the framework. I'm going to be addressing micro, meso, macro scales, same questions, different scales, different approaches. So the outline of today's talk is I'm going to give a little foundational information to kind of set up why I think micro is kind of necessary foundational knowledge to biogeography. I'll then pivot to the second part of my talk, which will be talking about pattern. And I'm calling these vertical fingerprints in biogeography. And I'm going to give 4 very quick case studies. And then I'll go into pattern and process and how they interact. And here I'll be going more of a macroecology. I'm already behind in time, so I might have to cut some slides at the end. But this is a familiar image, I think, probably to most people here. This is a species richness gradient at a latitudinal scale. The next slide here is a bird endemism gradient in the Andes. We know this because most folks in biogeography are interested in how species richness mirrors climatic gradients in space, and that is latitude, elevation. And these are the 2 real dominant gradients that we've focused on. And my focus during my PhD pivoted. I was really interested in rock climbing. I was trying to think about how can I do this in Southeast Asia when I moved to Singapore. Not a lot of rocks, but really tall trees. And I very quickly appreciated how steep the climate gradient is created by these 50, 60-meter old growth pterocarp forests. And so here's another gradient. It's local, but it's steep. We've put climate loggers from ground at different increments from ground to canopy. And what we found is for a 30-meter interval, so ground to canopy, 30 meters of forest, we have about a 2 degrees Celsius change in that space. And that same temperature gradient is 400 meters in elevation. If you look at humidity, it's about 11% difference from ground to canopy, and you'll need about 2,200 meters in elevation to get that same change in relative humidity. Now you scale this up to latitude, we're talking kilometers and kilometers and kilometers of space. So it's a very steep gradient. And because of that, you should see very abrupt changes in richness. At least that's what we think. And so that kind of set the foundation, just thinking climatically, okay, at local scale, I could take some of these foundational questions that have been talked about in biogeography at latitude and elevation and apply it quite locally. And so, again, it's this idea of thinking about latitude, appreciating patterns in elevation, latitude, and then also in height, and then coupling these. And what's really interesting is when you nest them, um, you can get um, kind of patterns that are responding in the same way, but in opposite directions. And that's because the climate gradients are opposing. So let me explain. So in a canopy, right, the top of the tree is hot, the understory is cool. Now we have these trees across elevation, but in elevation, the top of the mountain is cold and the bottom is hot, right? And so if you once you add trees onto the mountains and you look at climate, you have multidimensionality in climate space. So let me just show you what that would look like. So this is our traditional view of climate, right? We think about climate in mountains as like a layered cake, right? Each band is a unique climatic zone, and communities kind of assemble based on each layer of that cake. The kind of an arboreal view or vertical lens, when you put the trees on the mountains and you get that dimensionality, is that a climate regime in the understory and the lowlands is the same climate regime in the mid-story at a mid elevation or the canopy at higher elevation. So if you're looking at that purple yellow point there, that's essentially the same climate regime, but shifting dynamically in space as you go across elevation. So this sets up a system that allows for really interesting dynamic questions to be answered. So if animals, and just to showcase this, if animals try to stay at an optimal condition, how do they do this given this elevation versus height relationship that I just showed you? I've used this schematic a lot in my talks. It's just because I love this little green frog. So if you're this little green frog and you're living in the lowlands, right, and you really like warm, moist environments, right, as you move up to mid elevation, warm and moist occurs in the canopy. And so if you have the ability to climb, you can shift your vertical niche to track that optimal condition. If you're at a mid elevation frog and you appreciate cool and wet, as you move up, you can shift to the canopy to acquire cool and wet. And then there are these two kind of exceptions to the rule. That is a very hot, very dry canopy in the lowlands and a very cold, very wet understory at high elevation. And here, because there aren't any alternative gradients to exploit, it usually requires either a physiological or a behavioral adaptation to exist in these spaces. And I'll talk a little bit more about that in some slides to come. So why might arboreality serve as an important trait linked to biogeography? So I want to get into some foundational literature here. And so Jansen, a classical paper in tropical ecology is this paper by Dan Jansen, Why Mountain Passes or Are Mountain Passes Higher in the Tropics? And Jansen proposed this climate physiology model where basically, because of aseasonality and temperature in the tropics, you get that banding that I talked about, right? The lowlands are always different than the uplands in terms of the climate regime. There's very little overlap in space. They're disparate environments. And because of that, you get high turnover in communities as you move across elevation and as you transition from one band to the next. However, in a temperate mountain system, because of seasonality, you could be in the lowlands in the spring experiencing a climate regime and experience that same climate regime in the uplands in the summer, right? And so you can track climate in space. And so there's more overlap in space owing to time, seasonality changes in climate over time. And because of that, there's all sorts of predictions. I'll talk about the predictions that you can make because of that overlap here in one second. But what we wanted to do is just say, okay, well, if let's take Jansen's model here and apply it to a tropical forest. Can we do that? Because we know the understory is a highly buffered, kind of an aseasonal place, and the canopy experiences daytime fluctuations in temperature. And so the prediction here would be that in a tropical system, now we're not thinking latitude just in the tropics, thinking about a climate gradient vertically driven by forests, the soil ground layer would be akin to Jansen's tropical mountain, and the tropical canopy, because of high variation, a canopy in the lowlands will have some overlap with a canopy in the uplands. So the tropical canopy is akin to a temperate mountain, Jansen's temperate mountain model. We tested this. I did this during my PhD and postdoc. A wonderful PhD student of mine who finished recently, Dave Klingus, who's now he's at Yale and will be joining faculty at Rutgers University. You should really look into his work. He's doing really exciting work. Dave tested this at a global meta-analysis with a global meta-analysis and found tropical soils, because of the high buffering capacity, logs, soil, leaf litter, etc., very low thermal variation. The uplands and the lowlands in the soil are separated thermally. If you get to tropical ground, like the ground layer, the understory, there's a little bit of overlap thermally between the uplands and the lowlands, but in the canopy, there's high overlap in the thermal regimes because it gets really hot and it cools off quite a bit in the canopy. Right. And so that overlap there is denoted by the deep purple. This is a raw these are data from the Philippines. See Dave's global analysis and that for a meta-analysis. So, but here is why this is important. So if you have broad exposure, so you're exposed to high variability of climate, right? So high, hot, hot, cold, cold. Broad exposure should equate to broad physiological tolerance. If you can tolerate a wide range of temperatures, it increases dispersal or at least reduces movement costs, which would result in increased gene flow and decreased speciation, right? So that is the prediction. And so thinking about that workflow, exposure, physiology, dispersal, gene flow, speciation, and if where you live determines your level of exposure and by a climate, and if you live in the canopy, this is the four fingerprints here. I'm going to say canopy animals should have broad physiological tolerance. It should equate to larger geographic range size, reduced costs of movement, or at least lower turnover. So we had to use kind of a surrogate, so community turnover, so lower community turnover in the canopy over space, and greater niche lability. So, and that's a behavioral response there. So these are the four fingerprints of microgeography, and I'm going to provide some case studies on each of these to kind of showcase them working. So four case studies. This is the pattern part of the talk. So to first address physiology, we have to travel to Northeast Queensland, the wet tropical bioregion, where you have beautiful rainforests in the mountains and it flows right into the Great Barrier Reef. This is a really special place. I was very fortunate to be able to spend some time here. And during my postdoc, I was so tired of getting one frog per hour or two of work. I was like, I need to study something that is more abundant. And so I kind of gave up on frogs for a little bit and I switched to ants. And then I left, came to Florida, and I met this brilliant researcher, Lily Leahy, who I co-supervised with Steve Williams, James Cook. And Lily picked up and took what I did and made it brilliant. So to kind of first appreciate this idea of physiological tolerance. I just want to revisit a thermal performance curve just so we make sure we're all on the same footing here. So this is a classic thermal performance curve. You have body temperature on the X, performance on the Y. You could put any variable you want on the Y. On the left there, you see CT min. This is where the body temperature reaches a cold point where you're physiologically dead, right? So that's what we call a critical thermal minimum. And then on the right side of the curve, critical thermal maximum, the hottest temperature you can tolerate before physiological death. And so Lilly wanted to test, are there differences in the thermal tolerance limits, min and max, in ground and canopy ants, not only across elevation, but ground to canopy, right? So these two different spatial extents. So the first thing Lilly found is that thermal limits differed by elevation. So I'm hoping you can see my cursor here. CT max was always higher, right, in the lowlands. That's expected than the uplands. And the lowlands had a higher CT min, so they were less cold tolerant than the upland community. Second, Lilly found that there was distinct differences between ground and canopy ants. Um, Very nice. This is what we'd expect, right? So here's your ground community, your arboreal community, right? And so arboreal communities tolerate hotter temperatures and then also tolerate colder temperatures. And because of that, you have larger breath, right? And so they can tolerate arboreal ants could tolerate a wider range of thermal conditions than those living on the ground. Now, the effect of this is that it should translate over to geographic distributions, right? So if you can tolerate a wide range of temperatures and you can exist across different vertical niches, you should then be able to exist more broadly in space. And so the kind of the hypothesis here is that a broad vertical niche breadth, the ability to use different layers from ground to canopy, a broad breadth vertically, locally, exposes you to wide-ranging temperature regimes that then would translate to a broader geographic range size. And please please see Leahy et al. 2020 diversity distribution if you want any more information on this. What Lilly found was pretty much what we predicted, that as you increase the vertical niche breadth of a species, each dot here is a species, x-axis is vertical niche breadth, y-axis is the elevational range extent. So as species exploit more of that vertical space locally, it translates geographically to larger elevational range extents. The third fingerprint was that we should expect reduce dispersal costs. So movement should just be easier because you can not only tolerate more temperatures, you're exposed to wide-ranging temperatures, increasing your tolerance, and therefore you should be able to occur over greater space like we just showed. And so another way we could test this idea of dispersal costs is thinking about community turnover. So the gain and loss of species in space. And the idea here is a classic distance decay. So as you increase the distance between trees that we're surveying, you should have increasing dissimilarity among the communities, right? So the larger horizontal space across elevation, you're losing species and gaining species. So high turnover. So Lilly looked at the ground communities, compared them to the arboreal community. So if we look on the left panel of ground there, you can see that when trees are really close, low turnover, right? Low species turnover, and then a very quick upward curve, plateauing at maybe 700 meters in elevation distance, right? But so basically, the classical distance decay relationship applies to ground dwelling communities. However, if you look at the arboreal communities, very low turnover, so overall lower turnover and not a very steep change in community composition across space. Another PhD student of mine, Ed Basham, did a wonderful study on tree frogs and also showed this in Madagascar. His citation is down here in the lower right. Okay. And then there's some behavioral traits here that are at play. And so a lot of the kind of the fourth fingerprint is this idea of niche lability. So all of this can happen because these species are behaviorally plastic enough to adjust their vertical niche preference when needed. And so I'm just going to show two kind of studies that showcase this. The first was from my work in the Philippines where we did vertical surveys of frog communities from the lowlands every 200 meters all the way to the tops of the mountains. And one thing I noticed when I started my PhD was the classic understory species. So we'd be in the lowlands, we have an appreciation of what we're finding vertically, these understory species at mid elevations were up in the trees, and we're like, "What is going on here?" You think, "Oh, this is a ground species, this is an understory species, this is a canopy species." So that led us to on this whole path. And what we found is that individual species increase their verticality, they shift their vertical niche with elevation. So we're going to call that like a climate-induced niche shift. It's an abiotic response. I am still scratching my head on whether or not this is temperature or precipitation, moisture availability. I suspect in places like the Philippines, it's all about water, and temperature is relatively unimportant for frogs. That's so that's where I am right now, but we're always getting new information. The second case study here to think about niche lability was done a really nice study by a master's student of mine, Jesse Borden. He studied the native green anole to the southeast United States. That's that beautiful green anole here on the left. And he wanted to know what is the preferred vertical niche of this arboreal lizard. Well, when green anole is by itself, it prefers the understory. It's either on the ground or a few meters above ground. But there's another species, a competitor, a much larger, more aggressive species called the brown anole. This is a non-native species introduced decades ago from the Caribbean. When green anole shared space with the brown anole, so when they co-occurred, the green anole's vertical niche expanded 8 meters up into the canopy, and that is because the brown anole's preferred niche is right at the ground, right? And so this is what we would call a biotic response, where species induce this vertical niche shift. So those are the kind of the case studies, just to give you a little taste of vertical fingerprints and how they might play out in biogeography. Now I want to kind of pivot to pattern and process, where we're going to be thinking more from a macroecology lens, but also what is happening under the hood. So verticality, the fifth fingerprint, verticality is a system to understand global change. So a prior postdoc of mine, Bruno Oliveira, came in and he was really interested in macroecology. And we have these wonderful trait databases. Many of them decompose species at least a crude level of arboreal, terrestrial, and fossorial. And so Bruno looked at species richness globally of frogs. He then looked at at a community level, so the species per grid cell, compositionally arboreal, terrestrial, fossorial, and found just from this map here, if it's green, it's highly arboreal communities, that your arboreal communities tend to occur in the subtropics. And then as you go subtropical to temperate latitudes, you tend to get more integration of terrestrial and fossorial species. And you can see that here in this figure where the proportion of each strategy is represented across latitude. And if you look at the green, you can see as you move towards the center there on the y-axis, which is the tropics, you increase the proportion of the community that's arboreal. And then as you move towards the poles, the arboreal species fall out of the communities. And what's interesting is the arboreal and the fossorial kind of a mirror image. They're almost inverse of each other in terms of their relationship with latitude. We explored this a bit. We wanted to know what's driving these patterns across latitude. And what we found was if we look at this left panel of arboreality, so this is the composition of communities that are arboreal, that mean annual precipitation and vegetation complexity drove arboreality. However, temperature variation, whether it be seasonal or diurnal, drove fossoriality. And so in this situation, it looks like fossorial and arboreal communities kind of assemble by different rules, right? So the fossorials are all about mitigating, reducing climate exposure, right? They're all about living underground. So climate instability is driving fossoriality, whereas precipitation and habitat complexity drives. So you have to have habitat to use, and it has to be wet if you're a frog to live up in the canopy. So, and this is a high exposure strategy, right? But the conditions are right. And so exposure doesn't matter. But that was a global analysis, and it was a macro ecological approach. When Ed Basham, I mentioned him earlier, a PhD student joined the lab. He did a lot of the work in Madagascar. He worked in Costa Rica, Panama, Gabon. Ed is a field ecologist at heart. We started talking about the Oliveira study, and one assumption that I think is underappreciated. If you're a tropical ecologist, you probably don't. Uh, Underappreciate this, if that's the right way to say it. But if you don't work in the tropics, you might just assume temperature drive systems, right? And the tropics is aseasonal, but the tropics is highly seasonal. It's just seasonal in precipitation, right? Water availability comes and goes, temperature stays the same. And so Ed got really interested in this, and we asked, okay, if you're in the tropics, we know there's seasonality and precipitation. So does this mean that if we kind of blow out, if we go local, sorry, if we go local and we look at the verticality of these assemblages, do they stay arboreal, or is it more dynamic than we think? Right? Are we missing some things from the macro approach that we can appreciate at a micro meso scale? So if you see that, so to address this question here, we have to travel to Panama, where Ed did some really nice work from a wet season to dry back into wet, and then looked at the verticality of amphibian assemblages over the rainfall season. What he found was a really nice stratified species-rich community during the wet season that's showed here in the upper panel. So this is your wet season. You have height on the x-axis, richness on the y, and every color is a species at that height, right? And so you can see a very stratified community vertically. And then in the dry season, you could see that there's this collapse in complexity, right? There's a downward shift, fewer species, and those species tend to occur right at the ground or within the understory, right? So there's a compression in the community when you remove water from the system. Um, He simulated the data, so I'll just show you a few examples. So here's a Pristi mantis species. This species here shifted by around 11 meters. So how you interpret this is if it's negative, it's a downward shift as you move into the dry season. So this species shifted downwards towards the ground by about 11 meters. This poison dart frog is almost biphasic. It's amazing. It is entirely in the canopy during the wet season and entirely on the ground during the dry season. And then as a total community, overall, there was a downward shift of about 6 meters towards the ground. So, yes, the tropics are dominated by arboreal species, but it depends on what time of the year you visit, right? If you go to Ecuador or Panama during the dry season, you might find everything on the ground. But if you're climbing trees in the wet season, you'll undoubtedly find frogs up in the trees, right? And so it's all about the timing of the year. So what this is adding is a really important temporal dimension to this already complicated spatial dimension. So with all this shifting, just kind of I like schematics and I like this little green frog. So so just thinking about this, right? If you have this downward shift, you have this possibility of niche inclusion, right? So all the species shift down, you get a compression, all the community is hugging the ground, and things just expand, contract, right? These are short-term shifts. Perhaps there's periods of inactivity, estivation, where they just shut down and there's not a lot of interactions. But there could also be, fingerprint number 4 showed biotic exclusion in some situations. Competitors could exclude you from that downward shift. And this could occur when resources are limited at the ground level. And I'm going to show you some interesting data here, some results from an experiment we conducted. So Alex Becker, a PhD student, did a really nice study published a few years ago in Journal of Animal Ecology, and wanted to know, one, just fundamentally, do frogs just track water in space, right? So if you put water up or put water down, do they move and just track water? And then can we interrupt that tracking by injecting a biotic interaction? And I am almost out of time, so I'm just going to move a little quick through this. I do apologize. Um, we focused on two native species of frog, a green tree frog and a squirrel tree frog. We then had a competitor. So the green squirrel tree frog are competitors. We then added in a predator, which is the Cuban tree frog, which is a much larger tree frog introduced. It's a non-native species introduced from the Caribbean. We have an experimental design where we have these vertical enclosures with refugia from the ceiling to the ground. And then we had water treatments. We had a full water treatment where every single refugia had water available. We then had a ceiling treatment where water is only located in the highest refugia. And then we had a floor or groundwater treatment where just the lower refugia had water. And then we did individual runs at the species level to identify the basal preferred niche, so no interaction. This is the abiotic run, and then we had an interactive run, and so I'll provide some results on that. So, oh, this was my attempt this morning to simplify the next figure, and I forgot to delete it. So let's just go to this one. I've struggled with this figure for some time, and then I realized that this is actually just a lot easier than my attempt before. So the way we interpret this is every panel is a species. Okay, so under the full water treatment, green tree frog, squirrel tree frog, and Cuban tree frog preferred. Oh, and sorry, I forgot. So the start of the experiment, we placed the frog on this middle perch, and then we monitor the frog over time, right? And so interpreting this under the full water treatment at 0 is the perch. It's shifted slightly. So the green tree frog has a slightly higher vertical niche preference than the perch, which is in the middle of the enclosure. Same with the squirrel. However, the Cuban actually prefers kind of the ground, right? So it moved down from the perch. Under the low water treatment, green tree frog shifted down towards the ground, squirrel shifted down, and then the Cuban maintained its preferred niche because it didn't have to move to track water. When we look at the upper water treatment, the ceiling treatment, species now shifted up from their preferred niche, right? Both the green squirrel and Cuban tree frog shifted up. So essentially, this is just confirming, yes, frogs track water, right? They will respond. Their vertical niche preference responds to water. We then crossed each species we created a biotic interaction. So again, all green tree frogs, squirrel, Cuban. Here, the green was crossed with the squirrel tree frog in blue and the Cuban in red, right? So we see here under the green shifted down when it had a biotic interaction. It shifted up when it had a biotic interaction, and it shifted down again with a biotic interaction in the ceiling water treatment. Squirrel did the same, shifted away from the basal niche. These frogs represent the preferred niche placement, right? So essentially, to summarize this, is that adding a competitor or a predator triggered a response away from the preferred niche. It interrupted their tracking of water. So, in conclusion, forests provide steep vertical climate gradients. I provided kind of some evidence of these fingerprints, these micro-scale trait-based responses that are really important to understand to scale up to appreciating biogeography. Verticality plays a role in community assembly, right? So this, if you want to call it a trait or strategy, has effects in the landscape. Depends though, what scale, spatially, temporally, the interaction of space and time, and what taxa you're working with. And we have a bunch of work showing this is kind of taxa specific, and you get nuances. There's always nuances in ecology. Water is critical in facilitating the vertical niche, especially for amphibians. And species interactions can drive shifts in the vertical niche preference, specifically away from the vertical niche preference. Now, how might this play out? If we think about changes in rainfall moving forward into the future, right? That is a really interesting next step for research, I think. It's thinking about we show these short-term compressions in the verticality of amphibian communities. Um, if we that season of rainfall in the tropics, what are the repercussions, right? Is it going to be inclusion or exclusion, right? Or is it going to start interrupting the life cycle processes of species and therefore have a rippling effect in the community? And that is a really interesting question that we have not answered. If you like climate trees, I encourage you to pursue it. Okay, with that, I'm happy to take any questions that you might have.
Thank you very much, Brett. That was fantastic. There is some questions already in the Q&A box for the audience. Just FYI, you can always ask a question in an anonymous fashion if you don't want to put your name in there. I think that's a possibility in there, so feel free to do that. But let me read off the first one from Susan Renner, the president of TIPS. Why would an understory microclimate at low elevations equal an exposed canopy microclimate at high elevations? Light and temperature would be entirely different. And Brett, you should have access to those as well if you want to read it.
Oh, I would love to because I was trying. I'm a visual person, and so
you
I'm
should be able
helpless
to
unless I
open
can
the
see it.
Q&A box in there and see it.
I see webinar chat where I posted a link, but I don't see any other questions.
You don't see it at the very bottom by the share button. Should be Q&A.
Oh, I'm sorry. I am, oh wait,
All right.
not seeing it, but I'll just repeat what I think I heard.
I just put it in the chat now.
Okay, there we go.
So
Why would understanding at low elevation equal a exposed canopy microclimate at high elevations? Light temp would entirely. That is correct. I think the reason we're seeing this is solar radiation and surface temperatures. I think we've done a bit of work using infrared, and there's just more solar absorption in the canopy just via exposure, less shade. And so things just heat up. Um, and and you can get these superheated layers in the canopy regardless if you're in the lowland or the uplands. Um, that's actually it's a perfect question, and I could probably talk 30 minutes about this because it's really nuanced. At nighttime, heat escapes. Now it passes through the canopy, but there's also more wind. It cools off much faster. Um, yeah, I'm going to end there because otherwise I'll just keep rambling. But I think it has to do with, and especially in hot temperatures, it has to do with solar radiation.
Okay.
Shade.
Thanks, Brett. Apparently chat is disabled for the audience, so we are the only ones that can see anything in the chat at the moment. So our apologies for that. We'll make sure that the chat is enabled for everyone next time. But let me read the next question. This one is from Brittany Pooh. This was a really interesting talk. Have you thought about applying these hypotheses to plants themselves? For example, how does verticality of habitat and elevation determine range size, lability, vertical niche, etc. in plant species? It would require reframing due to growth stages and use of mean or max height traits, but it could probably be done and would be really interesting.
Now, just to clarify, and I'll answer this thinking about plants in terms of epiphytes, so things that can shift vertically, not just the ground-based trees or understory layer. For epiphytes, it's a wonderful question. I have a student named Lydia Soifer currently in the lab, Soifer, and she was totally interested in this and worked on bromeliads in Panama to see if some of these patterns I showed you play out with epiphytes, and they don't. They kind of do. It depends on the species, but there's a whole new set of rules that govern epiphytes. Um, so again, I apologize that I'm not gonna that is it is nuanced and it is taxa specific. In my conclusion I said taxa in there. It varies by taxa, even though I know I just talked about amphibians, but it is very much birds, totally different, reptiles different because again, reptiles actually want it a bit dry depending on where you are. And so I encourage you to take these questions and apply them to plants and improve everything that I published to be incorrect or at least taxes specific. That'd please me greatly. And we're already finding holes in everything I talked about. You know, so this is a narrative. Don't take it as fact. It's suggestive. It's evidence.
Thanks, Brett. Next question from an anonymous attendee. Put it in chat for you. Does vertical geography / microgeography apply to plant distributions in the Philippines?
So observationally for bird's nest ferns, the epiphytes, the dominant epiphyte is the Asplenium bird's nest fern. Observationally, yes. Empirically, no. So I know in my heart of hearts that I observe vertical shifts in Asplenium fern. Um, they they shifted higher up into the canopy, and I think it was due to water. They needed to dry out. It's too wet, and I think that's the same with frogs as well. It's just too wet. Um, but Lydia did the analyses on the data we collected and did not find it. But I know, I observed it. It's just the data didn't show it, and so we just have to go back and just more. I think it's more data. We just need more data. Um, So I'll leave it there. Not yes, based on my opinion, no, based on the data.
Okay. Next question from Andrew Villanueva. Really interesting data on seasonal vertical range compression during dry season. Is there evidence that biotic interactions may be stronger during the dry season? More competition or predation, perhaps?
You're spot on with the question, but as far as I know, there are no data to it's a it's a tough. We've been wanting to, that's like a next step. I just haven't gained the courage or found the right student that is interested in that is a high risk PhD chapter, in my opinion. So we have not pursued that. You should do that. And if you're interested, send me an email and we can talk on how you might do it because I have some ideas.
Okay. Another question from an anonymous person. How are these hypotheses you have tested with frogs and ants? How can they operate in large vertebrates like large bats and forest raptors? Do you think they hold?
Um, Probably not. Probably not. And that's just because if we think about local vertical niches like a home range size, like the unit of operation and the body size of like a large raptor, kind of they just birds are a bit they're a bit difficult for this theory. I've played with this a bit on how to generalize this across other taxa, and it gets just really messy, especially with birds. There are now, if you look at a paper I published with Steve Williams, it's Scheffers and Williams, I think it's 2018 in Frontiers in Ecology and the Environment. We actually play with this idea of how it applies to other taxa, and we provide some evidence on where it does apply for birds and reptiles and where it doesn't. Um, Lynn Schwarzkopf had a student do this in Australian reptiles, and it did hold in the savanna of Australia. Send me an email, I could forward you the paper.
Okay, great. A question from John Hart. Do these patterns play out for the prey species of the frogs?
I had a similar question asked to me at the one of my at a, I was given a talk at a conference, and I fumbled on. I couldn't really respond to it. Um, well, okay, sorry. You asked about the prey species and whether the prey. So now ants. I don't believe Lilly found a vertical shift in the vertical niche of specific ant species, or maybe it was really nuanced. Um, I know that Louise Ashton out of the UK had a really nice paper, I think, on moths that played with the vertical by mountain by latitude distributions, and I don't think they found a shift in the vertical distribution. Um, Again, I think it's taxa specific.
Okay, lots of questions. I'm just gonna keep going because we have 5 minutes left. This one is from another anonymous attendee. We are using ecological niche models to evaluate climate vulnerability. How would you integrate this other dimension of forest height in this vulnerability analysis?
Mm. Um, that is a really good question. It actually was a question that was posed during the last seminar, the last Vicky seminar. Somebody asked Brian McGill, how can you add a dimension so essentially what you're doing is you're taking the BAM diagram, biotic abiotic movement diagram, that's kind of foundational to conceptually for a distribution, then you're adding a dimension to it. And Lydia, sorry for the student who's currently in the lab, we've talked a lot about this, and it actually is a bit of a head scratcher if it's useful. Like, are 3D models useful? And what would they look like? And what questions would you address? I think they're interesting if you essentially you'd be time slicing as ecological models in time. If you did that, it'd be really interesting because you would capture some of so it's kind of akin to like saying, oh, I'm running an ecological niche model on neotropical migrants, but you don't account for the breeding range versus the overwintering range, right? And so it's very similar to that. You would be thinking about the drivers of verticality and then adjusting your approach to try to capture that temporal component on um, the marine world is a little bit more advanced in this than the terrestrial world. It's just way more, there are more data.
Mhm. Okay. Well, another one. If a broader physiological niche in arboreal species leads to larger elevation ranges, then won't it produce lower species turnover across elevations, regardless of dispersal? Can you separate these hypothesized effects?
That's if I let me see. Let me just make sure I understand the question. If broader, that's what we found. If I understand the question, that's actually what we found. So the high values on the y-axis and fingerprint number 3 were high turnover, and the uh, low values on the y-axis was low turnover. And so the ground communities had really steep, abrupt distance decay patterns, whereas the arboreal had kind of a flat line, intermediate levels of turnover. So it's just kind of like smooth and steady, but very shallow, right? It was just kind of the like constant turnover in space. And so I think, and then Ed Basham in 2018 published a paper that showed the exact same thing to support that question. So I don't think the hypotheses have to be parsed out. I think they support each other, if I understand correctly.
All right. There's another question about Southeast Asian amphibians. This person sees arboreal amphibians having narrow elevational distribution, contrary to the ant example. Can you talk about that? I'm not sure what study they refer to and if you're familiar with it, but
Um, Arboreal amphibian having narrow. I feel like we've talked. I feel like I've talked to Ed about this. So if that is the case in Southeast Asia, one one possible kind of in this is kind of a and I'm not sure what study it's being referenced. So I'm not suggesting I doubt this statement. It's just I don't have the knowledge. Um, is that it's more it's a higher energy expenditure to move for an arboreal species, right? They live in a multi-dimensional space, so they're moving up and down as well as across. And so movement, although they can tolerate more, this is the counter narrative to what I put forth. Movement should just by steps be more costly. However, I did have something in my PhD that I really wanted to play with, and this person here should take this and test it. And that is, there's a lot of parachuting in Southeast Asia. And I always wondered, is it because of emergent trees in its an energy model? So if you can climb to the top of the tree and parachute, you effectively get a hypotenuse, right? And so you actually cover more distance via the hypotenuse by gliding versus down and across. And so you actually save tremendously on energy by gliding. But I could not talk about this for Southeast Asia. It could just be movement costs energetically. Um,
okay.
and yeah, and just the if you're living on the ground, it's just ease of movement means you just traverse you traverse farther distances more effectively if you're living on a single plane versus multiple planes. So.
Thank you, Brett. Unfortunately, we're out of time, so I really apologize to those people whose questions we just could not get to today. But I'm sure Dr. Scheffers wouldn't mind if you emailed him your question.
My email is in the lower left
Yes,
on the
yes.
slide.
So please do so if you are interested in getting more answers. So thank you all very much for joining us, and thank you, Brett, for being willing to give this fabulous talk. It was really great. A lot of people have tuned in for your lecture. It will be posted on our YouTube channel if you want to revisit it in the future. And please join us next month for a lecture from Dr. Laura Pollock from McGill University. Thank you, Brett, and thank you everyone for coming. See you.
Thank you. Thank you all.